Hello, and welcome to the lecture about analysis of crystal surface reactivity by using interferometry microscopy technique, vertical scanning interferometry. My name is Cornelius Fischer. I'm heading the Reactive Transport Department, the Institute of Resource Ecology, and we are part of the Helmholtz-Zentrum in Dresden-Rossendorf, Germany, and we are focusing on reactive transport processes. Reactive transport, that means we have the interface of solid materials and fluids, and there is some reaction, there is some interaction, there is some dynamics included. So reactive transport can be observed by using experimental analytical methods, numerical methods, and today we are focusing on a very specific analytical method. It's a method that is able to capture the surface reactivity of solid materials. Here's an example. On the right-hand side of the slide, we have an XY plot illustrating the rate, reaction rate, dissolution rate of calcite dissolution as a function of pH, something that is quite often shown in a textbook. And there is the usual approach. If you lower the pH, rate goes up. If you go to higher pHs, above pH 5, you end up with a more plateau situation. But that's just half of the picture. If you dig a little bit deeper, and if you go beyond the classical powder experiments, if you analyze material that is more realistic, similar to that what is available in nature, single crystals, large single crystals, polycrystalline material, identical chemical composition, you end up with a large variability. And the rate spans a range of 2 to 3 orders of magnitude. That's an issue because parameterization of this range is pretty difficult to capture in reactive transport models. Usually we just plug in a single number, that's it. Nevertheless, our predictive capabilities are quite limited if we do not consider this large range, 2 or 3 orders of magnitude, for longer periods of predictions might be quite significant. So this compilation actually illustrates that the energetic landscape of the reacting solid material is much more variable than that what we usually apply in the parameterization of numerical codes. So we need data sets that are able to reflect this kind of variability. And this is done with surface sensitive methods. On the right-hand side of this slide, we have topographies, topographic maps of materials shown. I guess most of you have seen something like that already. It is reminiscent to the surface maps that are captured by using AFM, atomic force microscopy. The method I would like to discuss with you has a similar spatial resolution with respect to height. So it's in the nanometer range, and if you tweak the system, you can go even beyond, and you will end up in the angstrom range for height resolution. In contrast to AFM, the field of view is usually much bigger, hundreds of microns up to millimeters, but with a very high precision and height. So this provides the opportunity to study large realistic surfaces during reactions. And with this example, again, calcite, similar to the XY picture in the previous slide, we captured the evolution of surface topography with this method after reaction time of about 8 hours. Now we have the upper part, pristine surface, and then inert mask that serves as a reference, height reference. In the lower section, you see that the masked surface section did not react, but we have a surface retreat of the reacting non-covered part. So how is that measured? The technique, vertical scanning interferometry, is a technique that is reminiscent to the Michelson interferometer that you know from your physics textbook. You have a light source, beam, light beam goes to the beam splitter, the central part of the left-hand scheme of the slide. And after the beam splitter, we have two light beams available. One of them goes further to the right part to a reference surface. The second section goes down to the sample stage where our reacting surface is observed. That sits down there. Both surfaces reflect the light, and the reflected light goes back to the central part of our setup, and this optical setup enables superimposition of the two light beams. As a result, we see an interference pattern of each pixel of the object that gets observed. So if we provide height scan of the topography, all surface sections of the sample surface are at one point in the situation that we end up with a fringe pattern as it is shown on the right-hand side for each XY pixel. This gets analyzed in a detector CCD. So a computer stores all of those fringe patterns, and afterwards the envelope, this orange colored line, is calculated. And from that envelope function, we get an information about the specific height differences of each pixel. So we end up with a very precise height map. That's what is shown here with this example. If this height map is measured again and again during reaction at the same position of the material, we can take advantage out of this series of height information. This brings us to the surface reaction rate, what is actually an expression of surface reactivity. Usually with a rate equation, as is shown in the upper right, rate equals a so-called rate constant, then we have a so-called reactive surface area and a term that refers to the thermodynamic constraints. The problem that we have by using that, usually we do not have a spatial resolution to get the rates pixel by pixel, but the more important difficulty that we have is that the so-called reactive surface area is unknown. It gets modified over time. I have seen these height differences. There's incremental height evolution, so the overall area, surface area evolution is unknown. All these kind of calculations are something like a best guess with an unknown reactive surface area, and we come up with a mean rate constant that is actually an estimation of what's going on. With the technique that I discussed right now with you, we can circumvent this problem. And we get height differences at each XY pixel. Height divided by reaction time will end up with velocity. And this reaction velocity, the height retreat, or the material flux, these tiny little towers of each XY pixel, if you divide that by the molar volume of the material under consideration, you end up with pixel-specific rate constant. It's again calcite, no, the molar volume of this material, so we can calculate the rate map. And this is shown on the left-hand side. So all these colors that are shown here tell you that this chemical identical composition of the material that reacted here shows variability of its reaction rate. And you can identify certain sections with elevated or lowered reaction rates compared to any mean value. Now, with a classical parametrization, we would end up with one single color. Of course, you can use statistical methods. You can analyze the significance and importance of specific rate contributors And on the lower right, you have frequency distribution, and that illustrates that we have a multimodal contribution to the overall rate distribution of the material that is reacted here. With this powerful approach, you can gain a lot of insight into different types of reacting surfaces. Here's another material, another example. It's again a single crystal. I do not tell you what kind of material it is. Maybe you get attracted to this pulsating stars, the sky situation, and you try to figure it out. So what it tells to you is there's a large variability of surface reactivity, the energetic landscape that is responsible for this kind of material property provides very interesting constraints. If we would have the movie of this kind of overview available, you would see there's a pulsating behavior going up and going down of the surface rates. What is actually kind of interesting if we discuss a coupling of dissolution and growth processes of reacting material. So I hope I have been able to provide you some basic insight into how to tackle the problem of variability of surface reactivity. What I have shown to you, this is all abiotic examples. And within the SurfBio project, I would like to invite you to think about how to transfer things to systems where we have constraints that are set by biological constraints. Cells might be included, material that gets released by microbial activity, and that might be responsible for a modification of surface reactivity. And I can tell you, with the technique that I tried to introduce to you, you might be able to identify this kind of interesting behavior of fluid-solid interfaces. Thank you very much for your attention.